This is Unit 3, Cell Cycle Part 3. In this video, we'll be looking at how the cell cycle is regulated and what happens when that cell cycle regulation fails. The last video, we looked at the 3 checkpoints with concerning the cell cycle, the G1 checkpoint, or the point of no return, the G2 checkpoint, which says, is everything ready to go, and the M checkpoint, the third checkpoint, which is, are all of the spindles or all the kinetochores attached to a spindle fiber? If so, begin in anaphase. And so that was the 3 checkpoints that we looked at. One important part of this also, it has to do with this protein called cyclin. Cyclin is a protein that promotes cell division. You can see here on this graph that cyclin production begins during S phase. Cyclin production begins during S phase, and this picture, I think, is a very good picture of this. You see the cyclin protein here. Remember, proteins have a shape. Their shape determines their function. Well, in this particular instance, cyclin combines with another protein called CDK, or cyclin-dependent kinase. Cyclin-dependent kinase and cyclin combine together to make this protein here, which is called maturation promotion factor, or promoting factor. And the way that this works is when cyclin and CDK are combining, MPF is building up in the cell. MPF is what actually causes the cell to undergo mitosis. How does it know when to start? Well, when that MPF is at a particular concentration in the cell, mitosis begins. This checkpoint begins, or that particular place in the cell cycle starts, and mitosis begins happening. And it will be produced. Here's another picture of this. There's MPF activity, and the purple line is cyclin. That MPF will continue to be produced until anaphase, at which point, once anaphase starts, once those kinetochores attach, metaphase is over, anaphase begins, then all the cyclin will begin to degrade. As you can see on this picture, it goes away. Cyclin-dependent kinase, which is an enzyme, sticks around, but cyclin is degraded, and so goes the rest of the cell cycle. If cyclin were to stay in the cell, then the cell would continue to divide. There wouldn't be any stoppage of that. There would be no regulation. So we'll talk about that in a moment. That could cause a problem in the cell. Another sort of regulation has to do with the kinetochore itself. This has to do with the third checkpoint at the end of metaphase. As the kinetochores connect with the spindle apparatus, there are a set of enzymes that are going to be turned on, and these enzymes are called anaphase promoting complexes. Complexes is just a word for a lot of things, and so these anaphase promoting complexes are going to start forming once the kinetochores and the spindle fibers hook up. When the concentration levels of APC reach a certain particular level, that checkpoint level, anaphase is going to begin. There's nothing inside the cell looking around to see if everything's attached. Everything is chemical, and so the chemical response is, okay, everything is hooked up, and now anaphase can begin. When all the chromosomes have been attached to the spindle, it's ready to start anaphase. So just look at the words. Anaphase promotes the start of anaphase. So now let's look at some other ways that cells are regulated in their growth. Two of these things are called Anchorage dependence and density dependent inhibition. Density dependent inhibition basically means that a cell stops dividing when contact with other cells is made. So you can see this if you cut your hand, right? And that cut heals. How does it know to stop healing? Well, those cells that aren't touching anymore because you cut your hand, now they're touching again because that wound is healed. If they kept ill, or if they kept producing even after they were touching, you would just have this blob of cells, and that wouldn't be good. Anchorage dependence basically means that a cell must be connected to the connective tissue base in order to divide. So cells that are not connected to some sort of connective tissue will not divide. We have two layers of skin. We have the epidermis, which is the outermost layer of skin, and then the dermis, which is under that. The dermis is largely made up of connective tissue. And so because the epidermal cells are connected to that dermal layer, the dermal layer gives the cells food, and they allow them to feel. That's where all the nerve endings are and that sort of thing. Because they're connected there, they continue to divide. If they're not connected, then there will be no cell division. You can see this sometimes with a serious wound. If you lose, like, for lack of a better term, like a chunk of skin, from some kind of wound, that wound will not heal all the way back because that underlying connective tissue has been removed or damaged, and so that outer upper epidermal layer will not fully grow back. Again, if there's a problem with this, if there's a disconnection from the dermal layer and those cells continue to grow anyway, then there can be a problem. That brings us to the idea of cancer. Cancer cells do not exhibit anchorage dependence or density dependent inhibition. They will continue to grow even if they're touching each other, even if they're not anchored. Cancer cells are considered immortal in some ways, in that they'll just continue to divide. They don't stop dividing. As long as they have an oxygen supply, they will continue to divide. The body actually will make blood vessels to feed that new clump of cells, and the term that we often use for that new clump of cells is a tumor. It will create those cells for it. One of the first instances of being able of proving this was a lady by the name of Henrietta Lacks, and she went to a doctor in the early 50s, late 40s, and she was diagnosed with cervical cancer, and the medical team there harvested some of her cells, which are now known as Hela cells, and these Hela cells were found to be immortal. They were found to always continue to grow no matter what. They would grow in a dish. They would grow really well, actually, so they can be used to study cancer and how cancer is formed and what cancer causes. And so Hela cells, even today, have been used for a number of reasons. Even the polio vaccine was discovered largely in part because of the Hela cells that were available to Jonas Salk as he did his study. The sad thing about this whole thing is Henrietta Lacks was never consulted or never gave consent for the use of her cells, and it's a very sad story. The reason that her cells continued to divide was because of an enzyme called telomerase. And telomerase is an enzyme that adds telomeres to the end of chromosomes. And so if you have a picture of a chromosome here, on the end of all the chromosomes are these little telomeres. I'm putting circles, but they obviously aren't circles. You get the idea. Every time a cell divides, the DNA is replicated. Every time the DNA is replicated, one of these telomeres is removed. And so if there aren't any telomeres in the chromosomes, those cells won't divide, or they'll just die. And so what telomerase does is it adds telomeres to the ends of chromosomes, thus allowing them to divide indefinitely. This is particularly important as you are developing in the womb, and as you're developing as a young person. You're growing really rapidly. You need a lot of growth. As you age and mature, telomerase shuts down naturally. It's a good thing. Well, if there's a mutation that causes telomerase to continue to operate, then those cells will be cancers. A couple other terms concerning cancer. We've all heard these terms because cancer is very prevalent. Malignancy, this is when cancer is spreading. Is usually spread to other parts of the body for one reason or another. The other term that you're often concerned with is benign, or that you've heard is benign. This is a non-invasive type of cancer, usually not deadly because it's completely encapsulated. They'll go in and they can get the cancer out because it's all together in a ball or whatever, whereas malignant cancer tends to have fingers growing everywhere, as you can kind of see in this picture. It's not a good thing, and malignant cancer will often metastasize or spread to other parts of the body. Two main cancers that I want to are mutations that cause cancer. Again, mutations is a change in the DNA. Change in the DNA results in a change in the protein. A change in the protein results in a change in the way the cell functions. So ultimately, cancer is caused from a mutation in the DNA. Here's one such mutation. This is a mutation on the RAS gene. 30% of all cancers result from a mutation on this gene. This is where a G protein, which we'll talk about in the cell signaling unit, the G protein has been mutated, and so normal cell communication cannot happen. The cell cannot shut down the signal to grow that's going to the nucleus. That signal continues to be fired, and so it continues to reproduce. Normally that cell has this shut-off valve. In this particular case, there's not one because this RAS mutation, and the cell continues to divide. Another mutation that you need to be familiar with is a mutation of the P53 gene, sometimes called the guardian angel gene. 50% of all cancers result from this mutation. This affects a tumor suppressing gene. We have genes in our DNA that actively will kill cells that just will eliminate cells on purpose, and so that is a normal part of our body's physiology. And so if this happens, if this P53 gene is mutated, then a cell cannot self-destruct, as it were, and the cyclin that is inside the cell continues to be produced, and those cells continue to divide more and more and more. And I think it's important to point out that as we're dealing with cancer, mutations are largely random. There is a sense in which someone can inherit the mutation from their parents, but most mutations happen because of a random change in the DNA. Now, there are some things that we can come in contact with that cause a higher rate of those mutations, like anything that causes cancer, like we call these things carcinogens, like cigarette smoke or UV light or something like that. But by and large, cancer is a random occurrence.